Hi everyone, my name is Frederik Hammes and I'm a group leader in drinking water microbiology at IRM. In this presentation, I will give a general introduction into Legionella as well as an overview of the LeCo-Projekt. So let's just jump straight into the topic with a brief overview of some of the important aspects of Legionella as an organism. Legionella are gram-negative opportunistic pathogenic bacteria that cause severe pneumonia, while other clinical outcomes are also possible. This is a pretty large genus comprising over 70 recognized species and potentially many more. Of these species, we know by far the most about Legionella pneumophila, and information on other species is scarce. What we do know is that Legionella tend to prefer warm and stagnant aquatic environments. However, you also find these organisms in environments such as soil and compost. One interesting aspect about Legionella is that they have pretty specific nutrient requirements. Many Legionella are incapable of producing certain amino acids, and they tend to grow much better in the presence of iron. Partially linked to these nutrient requirements, Legionella inhabit biofilms and they proliferate in a variety of protists, to which I will come back in the next slide. Following their dispersal into the environment, Legionella is transmitted to humans through the inhalation of droplets. Legionella has a pretty interesting lifestyle. These organisms survive as free living cells in biofilms, but they multiply inside protists such as Vermamoeba or Acanthamoeba. What basically happens is that the protists graze on the biofilms, but once infected by the Legionella, these organisms hijack the digestive system of the protist, multiply, and are then eventually released into the environment, often to the detriment of the protist cell. This is, however, not the only possible outcome. There are examples where the Legionella can simply be taken up and persist in the protist without growth. There are examples where Legionella are taken up by the protist but are then degraded under certain environmental conditions or under certain species interactions. There's also been a recent publication that showed the interesting example where specific symbionts in the protist protect the protist cell against infection and multiplication of the Legionella. What is important here is that Legionella do not live in isolation in these environments. The biofilms provide nutrients for the protist, and they also provide protection for Legionella against stress and creates an environment where Legionella can multiply successfully. So where do we find Legionella in the environment? This figure on the right-hand side shows the complexity of the problem, in that Legionella are found in a wide variety of both natural and engineered habitats. This includes environments such as rivers and lakes, but also whirlpools, thermal hot springs, cooling towers, wastewater treatment plants, air conditioning units, multiple parts of building plumbing such as the boilers, your pipes, the faucets, shower hoses, toilet cisterns, and a couple of environments specifically linked to gardening activities such as garden hoses and sprinklers, potting soil, and compost. What is important here is not just the variety of possible environments, but also this one key point that for the vast majority of sporadic cases, the source of the Legionella is never identified. And this makes it extremely challenging to produce the correct guidelines and mitigating actions against Legionella if one is not capable of identifying the actual environmental habitat of the Legionella. In the LeCo-Projekt, we focus specifically on building plumbing as an important source of Legionella. Very often, these building plumbing systems create the ideal environment for Legionella to proliferate. This includes, for example, pipe materials that leach organic carbon, but it also includes warm temperatures and stagnation conditions, which allow for complex biofilms to establish themselves where Legionella can grow inside protists. Once released into the water, the Legionella can then be dispersed through droplets, for example, from shower hoses and shower heads to the end user, resulting in infection. Such an infection of Legionella can typically result in one of two clinical outcomes. Either the infected person experiences severe pneumonia, which is called Legionnaire's disease, and which can very often be fatal. The alternative outcome is called Pontiac fever, which results in milder flu-like symptoms with the infected person. When we look at a couple of studies that collected data on Legionnaire's disease during the last few years, it is very, very clear that the vast majority of Legionnaire's disease cases are caused by Legionella pneumophila. The one exception to this is the situation in Australia and New Zealand, where many of the cases are caused by Legionella longbeachi. It is also true that a number of other Legionella species have been associated with disease and clinical outcomes. However, these are more often associated with disease in immunocompromised patients as well as non-pneumonia clinical outcomes. Data on Pontiac fever is, however, severely limited. One really interesting aspect of Legionnaire's disease is that it is seasonal. There are approximately 3 to 5 times more cases in summer compared to winter. Why this is important is that if one can really underpin the reasons behind the seasonality, that may provide us with additional information on the major habitats of Legionella, as well as potential mitigating factors to control the organisms and ultimately the disease. The final important point is the global increase in Legionnaire's disease case notifications. This figure on the left shows the data for 4 different countries collected during the last 20 years. It is very clear that the reported incidence rate has increased globally in this period. What is even more important in the context of the LeCo-Projekt is that Switzerland has some of the highest reported incidence rates in the world. In response to this challenge, the LeCo-Projekt was established in 2020. This project focused on Legionella control in buildings and was supported by 3 Swiss federal offices. Although focusing specifically on building plumbing systems, the scope of the LeCo-Projekt was broad, with research questions covering technical aspects, environmental aspects dealing with buildings, ecological aspects, as well as epidemiological aspects. To cover this complex and multidisciplinary challenge, we divided the research questions into 8 different work packages and compiled a research group comprising of 5 different teams originating from 4 different research institutes with different experience and different expertise. Work package 1 focused on risk assessment, specifically from showers and shower events. Work package 2 and 3 were more technical, looking at standardizing sampling methods and detection methods, especially rapid detection methods. Work package 4 focused on the epidemiology and linked up with a larger Swiss-wide case control study, which is called the Swiss LeCo-Projekt. Work package 5 and 6 focused on buildings and specifically practical aspects such as temperature control, stagnation, and energy saving. Work package 7 dived into the ecology of Legionella and considered the possibility of using a probiotic approach against Legionella. Work package 8 looks specifically at chemical disinfection possibilities for Legionella. In this series, you have the possibility to listen to a number of presentations from researchers who worked on this project during the last 5 years. Tim Julian will discuss the infection risk from showers. Hans-Peter Fuchsen will talk about the establishment of rapid PCR detection methods, specifically within the Swiss context. Emile Silvestre also worked on PCR, but rather looked at the comparison between standard cultivation methods and PCR data. Melina Bigler and Daniel Moisenthal will discuss the role of showers as sources of infection. Dave Morgenthaler will talk about temperature control in building plumbing systems and specifically building distribution systems. Reto van Ooy will discuss the challenges for optimal water heating and storage systems design. I will give a presentation on the impact of stagnation and flushing on Legionella and Legionella control in buildings. Alessio Cavallaro will discuss the ecological interactions as well as potential probiotic strategies against Legionella. Marco Gabrielli will give a talk on Legionella diversity and the possibility of expanding the Legionella genus even further with more species. And finally, Antonia Eichelberg will give a presentation comparing different chemical disinfection strategies against Legionella. Of course, these presentations present only small snippets of the total project output. Eventually, a final scientific report and synthesis report will be released to the public. There have also been 18 peer-reviewed publications to date, and several more are in the making. The LeCo-Projekt resulted in more than 100 poster and platform presentations at scientific meetings during the last 5 years. In 2024, we held a knowledge exchange event where international researchers from across the world joined together in Switzerland to discuss important aspects of Legionella and Legionella control in buildings. There have also been public engagement events, including an open door day, as well as publications in trade journals. Education is an important aspect of the LeCo-Projekt. 17 bachelor and master students graduated from the project, as well as 4 PhDs, 4 postdocs, and a number of further education courses that were developed as a result of the LeCo-Projekt. If you have any further questions, feel free to contact me directly through the supplied email address or go to the question and answer section on the main page. I also strongly and warmly encourage you to listen to the other presentations in this series. That brings me to the end of the presentation, and I acknowledge the entire LeCo research team that works dedicatedly on this project during the last 5 years. I also acknowledge the funding for the LeCo-Projekt from the Swiss Federal Food Safety and Veterinary Office, the Swiss Federal Office of Public Health, and the Swiss Federal Office of Energy.